Hi, welcome to our webinar series on precision imaging agents. And today I would like to talk to you about the basics of CEST MRI contrast agents for cancer and kidney imaging. My name is Michael McMahon. I'm professor of radiology at Johns Hopkins and also at the FM Kirby Research Center for Functional Brain Imaging. So first, what is actually chemical exchange saturation transfer, or CEST? This is a switchable MRI contrast mechanism which requires a solute with a labile protons, such as this NH proton here, and it also requires applying a preparation pulse or a series of pulses in front of your typical imaging sequence. So when you place this saturation pulse on, what happens is it destroys the magnetization of the labile protons, and then through an appropriate exchange rate of these labile protons with water, this signal loss is then transferred to the water signal, which is the heart of MRI. And so the first papers which demonstrated these as MRI contrast agents were out of NIH, the University of Torino, UT Southwestern, and also Johns Hopkins. And this has started in around the early 2000s. One nice feature of these exogenous CEST MRI contrast agents compared to metal-based relaxation agents is that they're capable of encoding color to discriminate between the agents. So, for example, you can have a CEST agent with frequency 1, a second CEST agent with frequency 2, and a third CEST agent with frequency 3. And then when you move the saturation pulse across these different frequencies in the spectrum, you can determine what the signal loss is for each of these protons based on the saturation frequency. So this allows us to encode an artificial color by applying the saturation pulse and observing these water signal changes. And we felt like these would be very useful for a variety of applications, and first published our color CEST MR images back in 2008. So what people discovered is that cancer cells had a lot of fast exchanging protons, which they produced. And this is work by Peter Van Zyl and Jinyan Zhou back in 2003, and also Suzuma Mori. And so they developed these methods to study rapid exchanging protons using gradients to destroy the water signal and the WEX pulse sequence. And so they saw these cancer cells have these nice, very prominent amide protons with fast exchange rates, which was different between the cancer cells and the non-cancer cells. After this discovery, they then went into mouse experiments, and that was led by Jinyan Zhou, published in Nature Medicine. So what he did is, instead of applying the WEX sequence to detect these labile protons, he applied the saturation pulse on the labile protons, swept it across the spectrum, and he could show that you could highlight tumor tissue through this exchange contrast compared to healthy brain. In mice, this matched up very nicely with histology. And by the time 2006 came along, they had already implemented this type of imaging on cancer patients and showed that high-grade cancers were displayed particularly prominently. And that was published back in 2006. So it's a very fast translation of CEST MRI from tubes to mice all the way to humans within basically 6 years. And then after this, then there's been further attempts in cancer to decompose the water spectrum. And then what they found is, in addition to the amide exchangeable protons, they could also detect differences in amine and aliphatic protons for the rim of cancer cells of the tumor. And so this was published back in 2014, that not only would amide proton contrast display the tumors nicely, but they had these additional features. However, this is not a clear assignment to justify exactly what compound is associated with the amine, amide, and aliphatic frequency differences. So one of the first studies that I was working on with cancer and CEST was to try and understand what might produce these changes. And so what we had as an idea is we looked at non-malignant, benign, and metastatic breast cancer cell lines. And what we did is we took these cell lines, we lysed them, extracted the metabolites, took the NMR spectra of this metabolites, and then also compared this with the CEST Z spectra. And so we were interested in seeing whether or not changes in specific metabolites would lead to a change in the CEST contrast for these cell extracts. And so we had a couple candidate molecules which we looked at, and we found that glucose, glutamate, myoinositol, and creatine had very nice prominent Z spectra, and were also at high concentrations in these breast tumor cells. And some of the other metabolites, while they were prominent, they did not display a very strong CEST contrast. And so this was nice to associate with those four metabolites. We had another question is, can we design proteins for maximum CEST contrast? And this was based on the feature that to observe CEST contrast, the exchange rate must be sufficiently slow on NMR timescale so that the exchange rate is not so much faster than the chemical shift difference with water. And so you can see not all these labile protons on all the different amino acid side chains are appropriate. However, serine and threonine, asparagine, glutamine, and then the backbone NHs could be well detected, as could arginine and also histidine potentially. And so we then went ahead and used these expressions from Walter Englander's group, which related the primary structure of the amino acid backbone with the chemical exchange of the amide proton. So you can look at the left and the right amino acid side chains for a given amide, and then you can look at the acid and base catalyzed exchange based on this left and right amino acid, and then use their expression to calculate based on this empirical equation, which they described. And so we looked at a series of 12 amino acid peptides, and we came up with a list of 30, which would have very nice CEST contrast, particularly strong contrast in the case of the arginine-rich and lysine-rich peptides, with the signal amounts here, and then also the frequencies color-coded. So blue would be OH-based contrast, red was a guanylylamine-based contrast, and green was just an amide-based contrast. This is something we published back in 2008 as well. And we saw that, in fact, for the lysine-rich would have this type of pH dependence for the CEST contrast. So they were particularly well performing at around neutral pH. Based on this discovery, we thought about working on reporter genes. So what are reporter genes? We can place a gene which has some property which is detectable, whether through MRI, PET, or optical imaging, after a promoter to tell between live and dead cells based on expression of this reporter protein. And we can also look at cellular function, including looking at differentiation, secretion, and response with these reporter proteins. And so Asaf Gilad led this study where he developed this lysine-rich protein, which was a 200 residue protein. And he inserted this protein into 9L cells, which were also expressing GFP. And then when he looked at In vitro tubes of the cell lysates, he saw that in fact he could detect these proteins expression in the 9L cells. And in fact, also he could see elevated contrast for the tumors which grew in mice, which had this LRP expressing protein in the cells compared to the control tumors. However, after these initial studies, we realized we wanted much larger proton chemical shifts, particularly because we wanted to look for the optimal performing CEST contrast gate agents on clinical scanners. And what you see is if you go to high field and using, say, 4 microtesla or 6 microtesla saturation field strengths, you can see compounds with 1, 2 ppm quite nicely, large amounts of CEST contrast. However, when you go down to much like clinical field scanners at 3T, the optimum is above 4 ppm, and really, when you get to 9 or 10 ppm, you see extremely big amounts of contrast compared to even at, say, 4. And so we were looking for compounds which would show best. And one of our first attempts was looking at these ring protons, which had some extra electron density on the ring. And so thymidine is one such ring proton. And while natural thymidine doesn't display much CEST contrast, when we manipulate the ring by changing this double bond to a single bond here, or adding an extra methyl group at this position here, you can see that we get nice prominent chemical shift contrast at around 5 ppm from water. So while not ideal, it is definitely a big improvement over glucose and glutamate with this 5 ppm chemical shift. And this is something we published back in 2013. And then we discovered that we could tune it even further by having an intramolecular hydrogen bond, such as found in the salicylates. So in this case, what we looked at is if you just look at the phenyl OH, it resonates about 5 ppm and the exchange is extremely fast. The moment you put this carboxylic acid adjacent to the OH on the ring, you see a nice prominent peak at 9.3 ppm-ish, and the exchange drops down by a factor of 100 to about 500 times per second. And you can further adjust the hydrogen bonding partner for this OH, and you can also change the substitution of the ring and further tune this exchange contrast. In fact, here are a series of phenyl and anthranilate type of CEST agents, which resonate from 5 all the way out to 12 ppm, depending on the substitution of the ring and whether it's an NH or an OH. And so we've published a series of articles to describe how these phenol and anthranilate groups are really strong performing CEST agents at field strengths down to 3T. We also looked at imidazoles because it's another type of ring proton and with a lot of electron density on the ring. And so while regular histidine doesn't work and a lot of the azoles do not show much CEST contrast, when you have this nice extensively hydrogen bonded network through substitution at the 4 and 5 position on the imidazole ring, you can see these nice pronounced CEST effects at about 7.8 ppm for this ring NH proton. And here is just to show that the Z spectrum are very nice and very pH dependent, from 7.4 near ideal contrast, and then dropping down to near zero contrast, about pH 5.4. We also looked at some of the most extensively electron-rich groups in biology, which are the porphyrins. And so these NH protons for the free base porphyrins without a metal in the center can pronounce very large CEST effects at -10 ppm from water, with this CEST effect depending on the substitution of the ring, whether or not there are a lot of carboxylics, and also probably some structural effects on the ring, which can be transformed by having bulky phenyl groups or just aliphatic substituents along the porphyrin exterior. And so now we've identified a number of diaccest agents beyond the initial glutamate, glucose, and creatine. We have the iopamidol version, which the group in Turin is described quite extensively, the imidazoles, the salicylates, and these porphyrins. And so you can see that the chemical shifts span all the way out from -10 ppm to +12 ppm. So it's a quite wide range in frequencies that you can depict with the appropriate structures with CEST agents. And here's just a comparison based on some of the sensitivity plots for 11.7 and 3 Tesla, which we published recently. So now the question is, how can we use salicylic acid-based CEST probes for medical imaging? We'd like to test these particularly for cancer imaging. And so what I'd like to draw your attention to is aspirin, which is the wonder drug that nobody understands. Aspirin is this carboxylic phenyl compound here, acetylated salicylic acid. And so you can have this reactive group here, this acetylated group, and then when the acetylate comes off, that produces salicylic acid, which has this nice CEST effect. And so the action of the metabolism of aspirin into this salicylic acid turns on this CEST contrast. And this is interesting because aspirin acetylates COX-1 and COX-2 enzymes, and that's part of its mode of action. It also acetylates transcription factors and other targets too. And so this was something that was particularly interesting, to have a probe of aspirin metabolism using a new imaging contrast. And so one of the nice things about aspirin as well is that these are cancer chemopreventive drugs, and it's shown protective effects against the HER2 positive and HER2 negative breast cancers. And in fact, it's associated with a 41% reduction in luminal A breast cancer based on several studies. And so you can see that aspirin is a moderate inhibitor of COX-1 and COX-2, which results in an impact in prostaglandin synthesis and inflammation through this activity. And so here's just a cartoon to describe how salicylic acid, aspirin comes in, acetylates the serine 529 site, and then after this aspirin acetylation, this blocks this active site, and so the prostaglandins such as arachidonic acid cannot be synthesized. And so if you look at the CEST MRI spectrum for aspirin and metabolites, aspirin produces no CEST signal. Over the moment it's deacetylated, you can see this very nice big peak at about 9.5 ppm from water. And then if it's further metabolized through glucuronic acid addition, It then loses this 9.5 ppm peak, but then gains an additional 1.5 ppm peak from the hydroxyls and glucose. Similarly, we expect for the this other glucuronic acid addition to the OH site as opposed to the carboxylic site. And in this case of a salicylate, there's no CEST signal either between 9 and 10 ppm from water. And for this other metabolite where this 5 position is hydroxylated, then the chemical shift actually changes from 9.5 to about 8.5 ppm from water. So our first experiment was to inject 2 known COX inhibitors, salicylic acid and aspirin, and compare the kidney uptake in healthy mice. Here's our experiments. For salicylic acid, we saw some enhancement of the kidneys. However, if we injected aspirin, we saw much larger amounts of contrast by about a factor of 4 for the same dosage. And so use of aspirin injection into mice resulted in a larger amount of salicylic acid uptake in the kidney. And so that was one nice feature. We then looked at injecting aspirin into breast tumor bearing mice and see if we could detect this tumor uptake through CEST MRI. And so we looked at this triple negative MDA MB231 and also the SUM159 breast tumor cells. These were picked because the one is high COX expressing and the other is low COX expression. And here's our acquisition details. And you can see that we saw both nice amounts of contrast in the kidneys, and then also in the rim of the breast tumors, we saw nice amounts of CEST contrast. Here's an example of our CEST images for the kidneys and the tumor up to 60 minutes after injection. And so we saw about a 6% contrast in these orthotopically triple negative MDA and B231 tumor xenografts, which is about a third that seen in kidneys. So in summary, aspirin CEST MRI can be used to highlight breast tumors at the very least, with the rim of the tumor producing a higher perfusion compared to the core, which is typical for an actively growing tumor rim. So another feature of CEST MRI is that the contrast is often pH sensitive. And so this is a nice feature because it allows us to detect pH by observing the magnitude of water signal change. So if we look at low pH and we look at the CEST signal, there might be a 1 amount of CEST contrast. If we look at a high pH where the exchange rate is different, we'll see a different amount of CEST contrast. And combine this with the multi-frequency aspect of labile protons and CEST imaging, this is a powerful advantage for pH imaging. So one organ that is inconvenient for pH electrodes and these measurements should be useful is the kidneys. The kidneys tightly regulate acid-base equilibrium in addition to filtering out extra water and waste. And kidney diseases are quite common. It's greater than 10% of the US population has some form of kidney disease. And also acute injuries are frequently develop and need to be properly managed. And they're very difficult to characterize just using blood tests, which hampers decision making. I'd like to point out that in addition to MRI, computed tomography can be used to depict kidney function, and in fact, widespread use for abdominal imaging. In this case, we're looking at the CT images of a patient with a 7mm obstructing stone. What you see is in this iodinated contrast CT, in the unenhanced image, the kidneys are quite dark. In the corticomedullary phase, you can see that one kidney is highlighted more prominently than the other. In the excitatory phase, this kidney on the left side, which is actually the right kidney, is very fully highlighted, whereas this is still in the medullary phase for the right kidney in this image, but left actual kidney. And then in this delayed excretory phase, you can see that this left kidney here is cleared the agent, whereas this right kidney has not. And so this is showing that, in fact, there's a functional impairment for the one kidney compared to the other. And it's a greater amount of difference than you'd expect based on looking at just the collecting system and the size of that. There's not that much of a hydronephrosis. And so this is a patient where it wouldn't be expected there'd be such a big functional difference. And so what I'd like to present to you is that the imaging agent that we could use for CT, iopamidol, can actually be used as a CEST MRI pH imaging agent. And this is very nice because this imaging agent has been used in patients since 1986 with a lot of clinical experience. And in fact, it's also used for CT urography already. So all the pharmacokinetics for the kidneys are known. And so this agent is nice because it has two different types of labile protons, which are prominent at around 4.2 and 5.5 ppm from water. So you have this 5.5 ppm NH proton and these 4.2 ppm NH protons. If you look at the ratio of their signals versus pH, you can actually calibrate this. And so this signal ratio can be used to detect pH. Here's our pH map for a phantom of this agent for a wide variety of pH values. And as you can see, down to about 5.7 pH, you can see fairly nice pH measurements. And then below that, it kind of breaks down. And so here's a nice example of injecting iopamidol into the kidneys and looking at pH imaging. In this case, this is from the group in Torino. What they did is they clamped this right kidney here, the pedicle, and you can see a prominent drop in CEST contrast and increase in pH for this kidney. And then for this 20-minute clamp, you can see that the CEST contrast and pH values recover. However, if you hold this clamp on the pedicle for 40 minutes instead of 20 minutes, the pH goes a little bit further up, and it doesn't actually recover the pH or the perfusion of the imaging agent. So this is a nice technology where you can see, in this case of a acute kidney injury, you can see differences in the degree of injury through the CEST MRI pH measurements. Now, we thought of another application which might benefit from pH imaging, and what it is patients with methyl malonic acidemia. So patients with this genetic disorder are unable to convert methylmalonyl-CoA to succinyl-CoA, i.e. the body is unable to break down a variety of different amino acids, and this leads to an increased excretion of organic acid, methylmalonic acid, in the blood and urine. These patients experience a progressive loss in kidney function, leading to CKD, requiring transplants. And of course, so because these patients can't actually have a lot of protein in their diet, the creatinine is low in the blood serum of these patients because they have a low muscle mass, and so blood tests of the kidney function are not so good for these patients. So one of the things we did is we found a nice collaborator, the Venditti and Manoli lab at NIH, and they had a very nice mouse model where you can put these animals on a high protein diet and immediately turn on the MMA initiate the kidney damage. So if you hold these mice on a low protein diet for 2 months, their plasma MMA is low and their weight is normal. However, after even a month of being on a high protein diet, they actually develop very quick, large amounts of MMA in their blood. And then by 2 months on a high protein diet, they lose over half their body weight, which is nasty. And so we looked at a couple different groups. We looked at the unmutated control mice and the mutant mice on low protein diets. And then we also took these controls and the mutated mice on a high protein diet and compared these groups. And so here are just some examples of the pH maps we characterized on these mice. You can see for the unmutated mice on a high protein diet, pH maps were fairly uniform and about 6.5. However, if you put the mutated mice on the high protein diet, you saw a prominently reduced perfusion of the imaging agent and a reduction and also large variation in the pH across the kidneys. And so we could actually look at a couple different metrics which we measured in addition, the amount of contrast, the perfusion of the imaging agent, the absolute pH and the variation in pH, and correlate that with weight in these mice. And we saw for 3 of these metrics an excellent correlation of our measurements with weight, so that we thought this was a nice demonstration that we could use these metrics for looking at kidney function. So, and to summarize, CEST MRI is a new tool for molecular imaging with utility, and we've tested and identified a number of CEST agents with very excellent characteristics for 3T. We looked at aspirin for cancer imaging. We looked at iopamidol for kidney imaging. And we have a patient trial currently at our site for brain tumors, stroke, and have an IRB-approved patient study for administering iopamidol for patients with kidney injuries. So I'd like to thank all my colleagues in the FM Kirby Center in Radiology, Urology, Genetics, and also at other institutes and NIH for my funding. And thank you so much. And I'd also like to point out that we have a textbook on this topic. And if you have any more interests, please let me know. And thank you very much.